For Scott's fun fact. Okay, so another one I can't say without giggling a little. Scott had to hitchhike to his first oil spill outside of Houston. Upon arriving at the airport, he discovered he was too young to rent a car. Then his cab overheated in a traffic jam after a fire caused by the spill knocked out a bridge. An 18-wheeler picked him up on the side of the highway and delivered him to the command post hours later. His gag gift at the company holiday party that year was a cardboard sign that read, "Oil spill for us." All right, so Scott, the stage is yours.
True story. I'm going to talk about marsh fiddler crabs and periwinkles today, and I'm going to talk to you about 3 related studies from the Deepwater Horizon oil spill. Some of them were more response-based, and some were on the NERDA side, but we'll kind of expand out from there. But first, we're just going to kind of introduce you to the organisms. So this is the marsh periwinkle. It's a snail, a gastropod. The scientific name is Literary aerorata. Not much to look at, kind of, uncharismatic maybe to some, not to me necessarily. This is a shot of them in the field. They often climb on the Spartina stems and salt marsh habitats. And there's an adult on the right, and it's about 25 millimeters in total length. And there's a juvenile right here, about 4 millimeters in total length. Give you a feel for their size. Here's a shot, and some of you can maybe see kind of what they look like. This is maybe roughly about 1 meter squared. There's some snails in there. If you can't see them, that's where they are. And that's probably only about half of them. Some are probably covered underneath the vegetation you can't see as well. So this species is important. It's basically one of the few grazers in the salt marsh, especially from the intertidal and marine biota side. And they're not necessarily intending to graze the grass. They're grazing the microbes, microfungi, microalgae, things like that off the sediment surface and also off the vegetation. But when Spartina senesces, it gets really loaded up with microbes. And while they're in there feeding, they're also kind of shredding and breaking down that Spartina, kind of starting that detritus cycle that feeds a lot of the rest of the estuary, the energy source. They're also eaten by a lot of organisms that we kind of care about. 2 important ones. One would be blue crabs. So blue crabs come in and eat the smaller juvenile snails in particular and the subadults. And that's an important food source for them when they're part of the blue crab life history where they're in the marsh. And also diamondback terrapins, which is a estuarine terrapin or turtle. Conservation interests, are they state listed in Louisiana on the endangered or not? I can't remember, but they are in some locations. They're kind of rare, they're really beautiful. This is one of their primary food sources. So we'll jump over to the fiddler crabs. 2 main species in the Louisiana salt marsh. The one on the left is Gulf marsh fiddler, Euca longisignalis. It's kind of your typical dominant. So where the vegetation is really dense, where the soils are muddy and organic, you'll find these guys. The one on the right is a spined fiddler, or Euca spinocarpa. They're kind of like more open environments, not as dense vegetation. Sometimes they're on the edge of the salt marsh. They also like clay soils a little bit more. And you see in this shot in the background, some heavily vegetated areas of salt marsh. You'd see the Gulf fiddler in there. And then in open areas like this, you might see the spine fiddler. That's actually an area where some oil is still residual in that marsh. After several years, the vegetation hasn't come back. There's kind of an algal layer and clay soil right on the surface, and you'll find those species kind of where you'd expect them in that setting. So this is just a shot of the Gulf marsh fiddler in a burrow, and there's 2 males with their big claws kind of wrestling there, defending their burrows and trying to impress the lady crabs. I mentioned burrows because that's one quick way to kind of use a proxy to quantify crab abundance and size is what's the burrow density, and they're a lot easier to count than excavating the marsh and sieving all that sediment to count them. And also we look at the burrow diameter as a proxy for crab size. And this is just a shot actually from LumCon of Gulf marsh fiddlers in the marsh. Probably tides coming out. They're coming out on the marsh surface and getting active. They're real important through their burrowing activity. So They burrow down into the root zone of the plants. That helps flush out the sediments and oxygenate the sediments. Under some conditions, increased fiddler crab burrowing can actually increase plant productivity, increase the production in the marsh. If they get too high, they can also impact the marsh and result in erosion as well. So they really handle and process a lot of sediments. So they're important for nutrient cycling in the marsh sediments. They're also eaten by a lot of organisms that we care about. A lot of wading birds and marsh birds, clapper rails, herons, egrets, ibis really like fiddler crabs, as do things like redfish that we like to catch and eat ourselves. So important prey source, kind of just like periwinkles. This is how we sample. Oftentimes, it's not rocket science, right? It's a quadrat, and biologists out there getting kind of hot and dirty and eaten by mosquitoes and sandflies in the marsh, and we basically count burrows within a quadrat or count the periwinkle snails and other organisms. We also will collect them, at least temporarily, to do measurements on things like shell length or burrow diameter. And in most of these studies, we put the animals back and not collect them in the field. Sometimes for certain things we will collect, but most of these studies I'm reporting on today were not. They were just returned after they were sampled. So I'm going to start off with a study that kind of came out of the emergency response phase of the oil spill. It's really more of a field experiment. It's kind of in a somewhat limited area. You see that on the scale there. This is just a few hundred meters of shoreline, all on the same little island. And what we were trying to do here initially is figure out what were the best cleanup techniques. You know, what could we do to remove the oil from the marsh without damaging the habitat further? And so we had some oil controls where places got oil and we didn't clean them up to see what happened there. We tried different cleanup methods. When things seemed to work, we replicated those so we could study them longer term. And then in some cases, we coupled it with emergency restoration, where immediately after treatment, we planted those areas to see if that would help further. And we got kind of lucky in this case because right nearby, we had some areas that weren't as impacted, where the vegetation still stayed intact, that we could use as reference sites to kind of compare. That's what we were shooting for, trying to get back to. So kind of a small area, not a lab experiment, not a mesocosm, but a field experiment where we were manipulating things in the field in a small area where the oiling conditions were about the same. So this shows that shoreline in the oiling band. You see the oil wasn't necessarily as thickly back in the marsh, although it was somewhat exposed. This is what the ground conditions look like. So pretty bad. This is a good example of some of the worst oiling in the marshes during the Deepwater Horizon spill. So all the vegetation was dead, including the root zone. It laid over. It's kind of starting to harden up a little bit. And then underneath that oiled vegetation mat, there's about an inch of thick emulsified oil in the sediment. So you've got to think about, okay, maybe there's some toxic effects from the oil, PAHs in the oil, but there's also smothering and fouling effects under that level of oil loading. And both of those things can affect the plants and the organisms. This shot on the right here, the bottom right, is kind of what our reference area looked like. So impact versus reference. And these are just some examples of our treatments. This was our reference area, kind of what we're trying to get back to. This is manual cleanup. After about 2 years, just hand crews with rakes and shovels, trying to remove as little sediment as possible. You can't really see it in the photo, but there's a big patch of oil back here that didn't get quite removed, and there's oil residue all through here, but the vegetation was able to recover. This is a set-aside area where we didn't clean up, or an oil control, no cleanup treatment. A lot of oil on the surface is kind of weathered and crusty, but you dig into that right underneath it, there's that thick emulsified oil layer. The vegetation isn't really what you'd expect in there. This is not Spartina, and you can tell it's kind of stressed as well. Here's an area we treated with mechanical cleanup, both with and without planting. You see the effect that the emergency restoration had. That looks a lot better, a lot less oil in that area, less erosion as well, both lateral with shoreline retreat and also vertical. You can see that difference there between the two. This treatment got a little too aggressive, but where it was immediately planted thereafter, saw much better recovery, at least with the vegetation. So what were the animals doing? This is periwinkles, and if you can't see the slide exactly or read all the words, I'll always put a little picture of the organism. These are some dead oiled periwinkles. So each panel is a year. It goes reference, oil control, which is no treatment, mechanical treatment, treatment plus planting. And you see a big difference. So even into 2015, so like 5 years after the spill, still don't have full recovery. But you do see a bit of a trend with no treatment, and planting, where the combination of planting and treatment were improving things. And at least in this case, this is starting to get close to the minimum of what might be expected in reference areas. If you look kind of at a wider swath of Louisiana, kind of the lowest numbers you'd always want to see, it's kind of approaching that. So it's not recovered, it's in the process of recovery even 5 years later. So fiddler crab burrows, same story. We didn't sample in 2015 because we had some issues with water levels, but these are 2 years of data, same setup before. Here, planting made a big difference. And in this case, these two referenced and the planted were actually not statistically different, whereas the oiled and treated were. Planting made a big difference here. By a year later, the treated areas have caught up, but the untreated areas had not. Still lagging a bit behind, but getting closer to reference conditions. And lastly, in the same study, go back to these two species of fiddler crabs. So let's focus on this panel on the right. This is data from 2013. And what we started in their reference areas where there's all marsh, it was all strongly dominated by Longisignalis, or the marsh fiddler. And these areas where we didn't replant, we were either dominated by Spinocarpa, the spine fiddler, the one that liked less vegetation. The vegetation was not back, it was dead. There's kind of a clay layer on the surface of the marsh. This guy came in, so we had a species shift. He was either dominant or co-dominant with what we'd expect. However, where we went in and did that emergency restoration, that replanting, we shifted it back to what it originally showed. It's kind of an interesting result. We didn't exactly expect that. So now we'll jump from that kind of limited field experiment to natural resource damage assessment study looking at the injury side of things, where we try to look more widely. So now we're sampling across all of northern Barataria Bay, where a lot of the heavy oiling was, and our reference sites are spanning even wider, most of southwest Louisiana, to get that broader picture. We still looked at treated versus untreated versus reference. In this case, our treated areas were probably a little bit heavier oil to start with. And we also added something else to the study where we looked further back into the marsh. That first field experiment was just looking at the marsh edge. Now we're looking at the marsh edge, the interior of the oiled zone, sometimes 10 meters or so back into the marsh. And then we also looked at some areas just inland of the main oiling bands. There might have been some lighter oiling in here, but we're kind of looking for indirect effects, right? So if we impact 10 meters or more of marsh here pretty heavily, do we also impact some areas beyond? It's kind of what one of our study sites looked like. Heavy oiling, a heavy impact in that nearshore zone. Zone 2 is kind of back here near the edge of the vegetation, with a lot of oil still in the sediment, and then even further back beyond that, beyond the photo. So here's marsh periwinkles. Each panel here is a zone. So this is the marsh edge, oiled marsh interior, just beyond the oiling level. And we see effects for reference versus oiled in both zone 1 and 2. So the impact spreads further back into the marsh. And although the percent change here is about 90% versus reference, it's only about 50% here. But notice the reference numbers are a lot higher, right? So actually the numerical impacts were bigger further into the marsh, where the oiling wasn't even quite as severe and where the vegetation wasn't quite as affected either. So that's interesting. We also saw some size effects that I'll kind of skip over, particularly in the treated areas. So where we had a lot of oiling and intensive treatment, we lost a lot of the larger snails as adults were removed, and the recovery had to depend on new recruitment or movement into those areas, and those snails were generally smaller. On the crab side, we didn't really see in this particular study a lot of density difference. We did see some size differences, and we also saw that same species composition shift where the vegetation had been knocked back. And then we'll scale up one more time. So now what we did is we took all the studies we could find in the published literature, as well as some that were ongoing, folks that we knew who are sampling, and we looked at all that data combined in a meta-analysis for both species. So now we're looking at reference areas almost from Texas over into Alabama, oiled sites in Mississippi, again, concentrated in southeast Louisiana, but moving further to the west, and looking at all these different studies. So we have the data I already described, the ongoing sampling from that, NERDA studies, response studies, plus some Gomery studies, plus some NSF studies, McCall and Penning, Silliman et al., Dice et al., and combined all this data using a meta-analysis approach where we basically look at the ratios between the impacted sites versus the reference sites. And we analyze the ratios. That helps us kind of normalize for differences between the various studies and their study design and natural variation. Kind of mute some of that so you can look at what the overall results are. And this is the result for snail density periwinkles. And this is also set up by zone. We mimic that zone approach because several studies had done that so we could look at the bigger picture. So I'll start at zone 3. This is beyond the oiling band. No real effect there for periwinkles. Okay, it's similar to the Nerdist study, even across the wider area. Zone 2 of the interior of the marsh. We've got some impacts, and this kind of scale helps, right? If you ever are wondering what these indices look like. But we've got some impacts, maybe about 50% reduction, but within about 2 years, we're back to normal. If you're at 0, if you reach or cross this line, you're recovered for that particular parameter or metric. But zone 1 at the marsh edge, much more severe impacts, right? Almost 100% impacts, approaching 100% early on. Over time, there's recovery, right? So we're in a recovery process, but it kind of plateaus and tables off through 5 years. We're still at about, pretty good reduction here, more than between 50 and 90% reduction. Is this the new normal? Does this keep going? We don't know. There's some folks that have been continuing to sample a little bit longer. There's fewer long-term studies, so we can't look at it quite like this, but we are starting to do some of that. And density is starting to come back, but it's out near year 2018, 2019, and it's on density. Now, looking at size, do we have size effects? Impacts on, were snails smaller in oiled areas because the larger ones have been killed or because growth was reduced or recruitment was affected? Yeah, we did see that. It looked like it was recovered pretty quick initially at the marsh edge, but then it was declining again. And some other studies that have been ongoing have been seeing that. And even in 2019, one particular study team I know that I keep up with, their densities look recovered, but they're still lacking. They're not getting the larger snail back into some of the heavily oiled areas. We'll switch over to fiddler crabs. Same approach. Let's start with zone 3. So this is beyond the oiling band. Whoa, look at this. Indirect effect early on during the spill. Maybe they're more sensitive in some ways to indirect effects than periwinkles, but we got recovery within a year. For zones 1 and 2 in the marsh edge and the marsh interior, pretty good effects, not as strong, not as severe as for the snails, but still there. And even through 4 years, we weren't quite recovered yet. We were at about 50% reduction still. Now, I know that these came back looking at other studies within a couple more years. So, if we project this out, probably looking at 2015, so 5 years post-spill. Let's look at what size was doing. Impact on size in zone 3, beyond the oil, deeper into the marsh, beyond the oil, but recovered pretty quickly. Not much data for zone 2 in the interior. At the edge, also some impacts. We don't have any data from 2010, but we're still seeing it 2011. But by 2 years, leveled off near reference. Okay. And for the most part, in the studies that I know that are ongoing, and there's only one I can really think of that even just sampled this year, they think that they've seen recovery of fiddler crabs by now, but again, not for the periwinkles. Now, this is a little bit different take. This also came from the Nerdist studies that were ongoing, but it was looking at the restoration side. It's also a meta-analysis done by someone else, Matt Bauman and his co-authors. And while we were looking at injury, they were looking at, well, how long is it going to take for a new marsh or a restored marsh? You might do marsh construction or marsh creation or marsh restoration, kind of offset impacts from the spill under the NRDA. But they want you to know what's that recovery time or what's that developmental time for these organisms in newly constructed marsh. Why would you need to know that? Because your marsh that you restore might not be the same as the marsh that was impacted, right? You may need a bigger area. You may need a different area. But these things are starting from constructed marsh. You're kind of starting from scratch. On oil marsh, you've got to develop marsh. You impact it, you knock it back some. It recovers over time, hopefully. These marshes are developing often from new sediments that have been dredged up and placed in the intertidal, sometimes planted, sometimes not. So what did they see? This is development of this population in the absence of oil in a newly created marsh area. Most of these were new marsh construction sites. So it takes a while, at least 4 years till it approached baseline or reference conditions. But did it stay there and level off? No, it's kind of up and down, it's fluctuating. And people interpret this graph a different way. The author in this case said, well, you know, it looks like 4 years we reached reference. You might make a case, well, yeah, but you really didn't get back there until 7 years later, and you dip down again. This was actually not statistically significant from 0. So 7 years, you know, you might call this 12 to 14 years. I like to see kind of a curve, and they're going to fluctuate and then kind of level off. So, you know, I'm looking at recovery probably out here, or development of that population out here sometimes. So what's important about this? It can inform what's happening in the marsh. If you got a similar situation, you knock back the population of oil marsh this far, and you have residual oiling conditions, that might prevent that recovery. Maybe not. Also, in a nerdist sense, you've got to think about the injury. What did you impact, and how long does it take to recover? Then you have to look at the restoration side, right? What did you gain with your restoration? How long did it take for those gains to accrue? You have to kind of integrate those two to figure out how much restoration. That's part of what we do with NERDA. So just to wrap up, periwinkle impacts, relatively severe, especially in those heavily oiling areas. Recovery time frames greater than 5 years, even with emergency restoration, even with that planting. And for some of the studies that are ongoing, these populations still haven't recovered even 9 years later, especially in terms of their size distribution, their distribution of adults versus juveniles and subadults. Fiddler crabs, we still saw impacts there, maybe not quite as severe compared to the heavily oiled sites for periwinkles, but we also saw fiddler crab impacts in areas that had lighter oiling, it's just shorter duration. So recovery times there, 1 to maybe 5 years, maybe a little bit more than 5 years. Don't have as much data there. And then compare that to the restoration side, at least for periwinkles, where that meta-analysis has been done, comparing all studies over time. 4 to 7 years for those populations to develop. So we've got to integrate those two to figure out, you know, how successful is restoration in the long run, both for planning, but also for later assessment. And just to kind of wrap up with some ongoing work, there's some new studies that have come out, especially for grad students completing their master's and dissertations. So new data is available now. Some longer term studies have been ongoing and are still sampling. We're now bringing in more of the restoration component. Right now we've got a GOMRI grant where we're looking at combining all that data into a larger meta-analysis and updating some of those things to see when did we actually reach recovery and what are the restoration curves look like. So that's it.
Okay, great. Thank you so much.